Hello, everyone. For those who don't know me, my name is Jessica Williams. I am a sequence validator for the Fungal Diversity Survey and the Ohio Mushroom DNA Lab. What I wanted to do today was just have a little discussion on my philosophy around sequence validation and dig down a little into how I think about these things. So, if you are unfamiliar with what I'm talking about, we use a technology called nanopore sequencing to do high throughput sequencing of fungal specimens. We sequence hundreds of specimens at a time, and these sequences all need to be manually verified to make sure that they are, in fact, solid sequences and not contaminated or off target. We are not doing full genome sequencing here, but are doing what is called DNA barcoding, where we use a small chunk of DNA that tends to be relatively consistent within an individual species, but varies between different species. This, with some exceptions, allows us to identify species with just this barcode section. For fungi, we're mostly using the internal transcribed spacer region consisting of ITS1 5.8s and ITS2. I'll try not to get too technical with this video, but in brief, the way that nanopore works is by taking multiple reads of a single chunk of DNA, then forming a consensus file of these multiple reads that gives us our final sequence. This isn't always a perfect process though, so we have to check everything, and to check them, we have to compare them against the sequences that are currently in the various DNA databases available online. Sometimes these validations are easy. Some species have very consistent barcodes, are widespread, widely sequenced, and have agreement in the databases on what barcode represents a particular species. Turkeydale, for example, Trametes versicolor, is a very good example of this. It is a widespread, common species and has a very consistent ITS. Validating these is extremely easy. Issues can arise though with some sequences, and I want to talk through a few of these issues with you guys. And the main thing that I want to emphasize here is that with sequence validation, we are not describing new species. We are not trying to replace the publishing process. We will give temporary codes to things that lack solid reference data, but these codes are just ways to group similar sequences together so that when someone does go to describe a species or connect an already described species to a temp code, it's easier to parse through the data, right? Like when you have clusters of temp codes, it's a lot easier than looking at this wide spread of sequences all clustered under one name, and then you're not sure what actual sequence represents that species name. With that, then, my goal is to find the best name for things, given the available literature, reference databases, and the consensus of the mycological community. I'm not trying to break the system or cause a big fuss unless something is obviously incorrect. That said, though, there is a lot of very incorrect data in the various sequencing databases. There is little curation for the names that are attached to the sequences in databases like NCBI GenBank and others, and sometimes there is a massive disagreement on what sequence represents a seemingly common species. So we have to go at this with a bit of skepticism because otherwise we could be causing errors in the system to perpetuate. It's much easier to fix bad data if you catch it early on. So when I'm looking at a sequence blast, after I've made any edits and I know my sequence is good, there is an order through which I'm going to process things mentally. So if what I'm looking at is a completely novel sequence, i.e. nothing is showing above maybe 95% similar, those are going to be easy. For the ITS region, 5% is huge. There are very few individual species that are going to have that big of a spread in their ITS. This is usually a situation where I can confidently assign the temp code and just move on. If someone else likes, they are welcome to use that public data we upload to iNaturalist, MicroMap, and GenBank to try to connect it to a published species name or describe a new species. I'm not trying to do that. We don't usually have micro data on what we're looking at. We usually just have macro photography. And it's just not within the scope of what I'm doing to try to like connect old names to a sequence based on macro photos and micro. It's just not what we're doing. So a temp code will suffice. If it is still novel, but closer, maybe above 99% similarity, or maybe even above 98% similarity for some groups, that's when I have to start thinking a little harder about it. Like, what species is it matching? is that species part of a group that is known to have a lot of variation in ITS, like some hygrophorous species? Or is it in a group where a single nucleotide difference indicates a different species, like some Cortinarius? A lot of times, for this question, I'll end up taking a look at iNaturalist notes for close sequences and see what people are commenting. Usually, for the difficult groups, you will find some discussion on how much variation represents a new species. Looking at the morphology of specimens can help too, though you do need to keep in mind that some species are just variable. So Trametes versicolor, for again to use that as an example, is a very solid species concept that has a ton of morphological variation. So if you described a new species, every time you have a morphologically different turkey tail, you'd have a lot of different turkey tails that are all just still turkey tail, right? Like, so you have to keep that in mind. Danny Miller's website actually can have some good information when you're trying to figure this out too. It is specifically for the Pacific Northwest, but it does have a little bit of application outside of the region, and at the very least, it can give you good information on some of the more difficult groups. And I will drop a link to his website in the description of this video. If I get really stuck, there are experts on INAT that will respond to being tagged and usually will have some good information and are usually very willing to share their knowledge, right? Like scientists are all nerds. They all want to talk about their favorite subject. I am not going to call any specific people out here on YouTube, but if you are in the mycosphere on iNaturalist long enough, you'll quickly figure out who the knowledgeable people are. And it does help to check people's profiles. Folks will usually outline their areas of expertise. They want to tell you what they're passionate about, right? Use that information. Now, after I've kind of thought through this, at this point, if I do decide that it is novel enough to warrant a new temporary code, again, I can just assign it a temp code, explain my reasoning for adding that temp code on the iNaturalist page, and then just move on to the next observation. Where it can get really weird is if you do find one that matches or is really close to a named species. If you're really lucky, there might be a type specimen that has been sequenced that you can use as a reference. But even if there is something marked as a type, it's not always a guarantee. There are occasionally types that are mismarked in GenBank or on Mycomap that aren't fully trustworthy. So sometimes it is worth it to kind of look at the accession page for that type, see what the paper it is attached to is, see where the specimen comes from, make sure the type matches the type in like Index Fungorum or Mycobank, just to make sure it's legitimate. But if you don't have a type, and often you're not going to have a type, you then need to consider if the name it is matching is a good name. The first thing I will usually do here is pull up the NCBI blast page and blast that sequence against the name it's matching. So what this is going to do is it's just going to only blast it against anything that's been uploaded under a particular taxa. If every sequence under that name comes back as a fairly high percentage match, above like 99% usually, I can be fairly confident that there is good consensus in the community that the sequence matches the name. If there are outlying groups with much less similarity, say there's a cluster of about 10 with only about 95% similarity, that is when you need to start questioning which name is the correct name here because something's misnamed. Like something is correct, something's being called this name, but probably represents a different species. At that point, I'll usually go check and see where it was described from. If the species is described from, say, Europe, and all the close matches to your specimen are also matching European specimens, and the sequences that are only around 95% similar are all from, say, South America, you can probably be pretty confident that the South American sequences may have a wrong name applied. Alternatively, I'll look and see what the source of these sequences is. If one group of sequences is from a recent monograph by an expert in a group, and another group is from a group of botanists doing plant pathogen studies where they don't even discuss in the paper how they came up with the names, I'm probably going to defer to the genus expert's monograph in this case. Depending on the observation, I do sometimes dig pretty deep into these papers to figure these out, but even with that, I will sometimes hit a wall and I can't determine what the best sequence for that species name is. And at that point, I'll probably make a temp code because I cannot be fully confident that what I am looking at represents the species name in the strictest sense. So, if I have multiple groups of sequences representing, for example, Mycena hematopus, I might give what I'm looking at a temp like Mycena hematopus OH01. Basically, this extra code number is just telling us that this is close to Mycena hematopus. It could be the real thing, but I'm not confident that if it is or not. But if you are confident in your species name, you could just call it that, give it your justification, and move on. Sometimes you will run into sequences that simply cannot differentiate a species. Hemaloma are particularly bad about this, as are certain mushrooms in Strobilurus or Armillaria or anything in Pholiota section adiposa, just to name a few. There are more problem groups where ITS just is not a good differentiator. With these, if there are multiple names in play and all of the resources that I can dig up indicate that they cannot be separated based on ITS, I will usually leave the identification at section or genus or subgenus or whatever the lowest taxonomic rank I can get it to is, and then indicate that ITS is just not sufficient in this case to get the observation of species. Ultimately, I'm just trying to do my best to either assign the best name to a species that I can, or give it a good temp code so when folks studying these groups look at them, they can then have easy indications on where different genetic groupings may be. If I make a mistake, it's okay. The beauty of doing all of this on platforms like iNaturalist, where the information is publicly available, is that someone with a better information at a later date might be able to just go in, refine my conclusion, and correct any mistakes that I might have made. So remember that. And try not to get too into the weeds on overanalyzing every single specimen that you look at because it can always be corrected later. Of course, be skeptical, be suspicious, but sometimes a turkey tail is just a turkey tail. And my job as a validator is not to reinvent the taxonomic structure. It is just to try to make sure that the information that we are adding to the DNA database is as accurate as possible so that it can be used as a good reference database in the future. Hopefully, this made sense. Hopefully, it wasn't too much of a ramble. If you enjoyed this, please give me a like and subscribe. I do appreciate it. And if you have any questions, feel free to reach out to me. I will do my best to respond. I'll drop a few helpful links in the description below. And have a great day, and I'll see you next time. Bye bye.